Antonitis. He will be talking about villages and early trade along Letaba River in KNP.
It's on. There we go. Good morning, everyone. We're changing tack quite significantly since we're talking archaeology now. And it's been quite an eye-opener for me, sort of being at this conference, because as an archaeologist, we typically deal with people in a landscape, right? And it's really sort of refreshing for me to see what's being done on the landscape level, right, from the ecological side. But the talk we'll be sort of discussing is research that we've been doing in the Kruger Park on the Letaba River since 2021, and it builds off earlier research that was done in Kruger itself, looking at the earliest farming communities in southern Africa. Farmers have been part of the African landscape in South Africa at least 1,800 years. And we've been studying these communities at least since the 1980s, where we've had a really good sort of understanding of that these early communities of farmers are on the landscape by about 200 AD, and we've done tremendous work in terms of understanding the distribution of these first farmers into the savanna sort of ecotones of southern Africa. And we've nailed the chronology down fairly finely. And in general, what we know about these communities and how we understand them are that they were pastoralists. They kept domestic herds of cattle, goat, and sheep, smelted iron, used other metals as well, and then obviously the agricultural aspect as well, right? Sorghums and millets being the main sort of food-producing crops that these communities had, right? Work in Kruger itself was conducted from the late 1970s onto about 1986 was the last date, where a sort of regional survey was conducted to document all the archaeological sites within the confines of the park. Through the University of Pretoria, they identified around about 320 sites spanning roughly 1.5 million years of human history in this region. The aim, however, for this early research was really constrained to identifying the distribution of human communities in the past, as well as the chronology associated with these movements in and out of the park, right? And the result was something, well, this is one of the outputs. What you have there is the park over there and a timeline over there, and Andre Meyer was sort of able to delineate sort of various cultures, as he would have called it, who occupied the park during the last 2,000 years. And in most of it was survey-based research that was done, and then in a few places, limited excavations as well. And one of the sort of major excavation sites were what he referred to as LE6 and LE7, which is these Letaba sites, these early agricultural sites on the banks of the Letaba. And they basically stayed up until 2016, when one of my master students for a project really looked at some of the faunal remains that were excavated from these earlier excavations in the park. We'd known up until then that the utilization of wildlife was the predominant food procuring strategy for these communities, unlike communities outside the park who tended to have a lot more domesticate in the diet, right? These Kruger communities, and especially the Letaba communities, were almost exclusively reliant on wild animals for their meat requirements. But not just wild animals, not just any wild animals, a lot of, there we go, very large animals, and sort of in the sort of really sort of larger categories of animals. Whereas the picture outside the park is that there's a lot more snaring and sort of individual hunting taking place. So you tend to have a picture of a lot more sort of medium to small animals in the sort of faunal profile. This sort of necessitated a return to the sites, right? Because we lacked the spatial data to actually look at what these patterns that Evan identified, what it actually means chronologically, but also spatially, how this was sort of utilized at the village level. Unfortunately, this was the extent of the spatial data we had for the sites. Sort of a map that was drawn up in the 80s with a little bit of detail over there, but not much, right? And for archaeological purposes, I'm really talking about how the sort of village level dynamics would have worked and how we needed more data, right? So we went back, identified the sites, we managed to relocate them, and as far as we know, we were the first archaeologists back on these sites since the 1980s. And this is what the sites look like, right? So these open areas are all anthropogenic, right? This is human-induced sort of clearing. And as I mentioned, this is about 1,200 years ago, right, when this village was last occupied. As I said, it's on the banks of the Letaba. There's the Letaba, its first high terrace just above the southern bank. And the archaeology is all over the surface of the site, right? It's actively being exposed by erosion, especially on the edges of the site. There's a lot of sheet erosion taking place and the Donga Formation actively sort of eating away into these sites. And that sort of prompted us to conduct some rescue excavations on the site as well to sort of at least save some of this archaeology before it's washed away completely. But during the survey, one of the sort of most important finds that we were able to sort of identify were this object here, right, which is a blue-green glazed ceramic. Now, I'll return to that later on in the talk, but just take note of that it is significant. It just looks different, right, to what you'll tend to expect on in terms of what these locally produced ceramics would look like. Right, we started excavations, and not surprisingly, given the earlier research that was done on the site, lots of faunal remains coming out, right? Animals that were butchered and consumed at the site, and also managed to locate more of these blue-green glazed ceramics. And the great thing is we found it in excavations, so the context is good, we can date it, and we're also finding it in association with sort of low-fired earthenware, which is locally produced, as well as a variety of other sort of faunal remains. Most of the archaeology is located in these subterranean pits, right? It's not very deep, but it's pits that are dug into the soil, filled up with trash, predominantly animal bones and ash, and then covered up again. And so what you see there in profile, that lighted dot over there, that is one of these pits, right? And they're sort of semi-hemispherical in shape, and that's sort of the surface of a donga there, right? And typically these pits are filled with cultural remains, sort of evidence of slag, metal production, in other words, copper, iron, ostrich eggshell bead production, the odd vulture wings that we find thrown away as well, all manner of things, right, that these communities were actively using on a daily basis, right? But the fauna really is the one category of data that really stands out. And from what Evan sort of had started on, what we're showing as well is that there's a systematic and very intensive hunting industry taking place at these sites, right? This isn't just for food and for consumption, this is something much bigger, right? They're hunting large herds of buffalo, well, large-ish buffalo, really large individuals and we have the entire age profile of juveniles, sub-adults, adults, and old animals as well, right? So it's probably large hunting events that are being used to target these animals. What do we have? Giraffe as well, right? And not just one giraffe. We'll typically find 3 or 4 giraffe individuals sort of butchered at the same time. And this is all, as I mentioned, really quick hunting episodes. They're butchering the animals, they're throwing it away with a lot of meat still attached on the animals, right? And we see that in the fact that a lot of the skeletal portions are still articulated when they were thrown away, right? Which means there's some meat and muscle and sinew still attached to the skeletons. A lot of these sort of skulls that we're finding as well, that's a blue wildebeest, sort of the bottom part of it, but you can see that the horns were, for example, chopped off. You still have the butchery marks. So using these butchery techniques and marks, we're able to reconstruct the sort of rapidity and the techniques that were used in order to sort of process these carcasses. And just to sort of to mention these phalanges, this is almost a signature for these sites, right? These phalanges are split down the middle, marrow extracted and thrown away, right? And it's quite rare to see on the scale we're seeing it at this site, right? Just the sort of local ceramics that we're using to date it. But what sets this site apart from and really piqued our interest initially was the trade goods, right? So we have Asian trade goods in the form of glass beads, which, as far as we can tell, is probably the earliest evidence for Indian Ocean trade in the interior of South Africa. We have marine shells from the Indian Ocean, as well as these blue-green turquoise glazed ceramics that I pointed out earlier. And these objects, what these ceramics would have looked like if they were sort of complete, would have been one of these vessels over here, right? And these are incredibly unique within an African context because they are made in southern Iraq, right, in the modern town of Basra, between the 3rd and the 10th century AD. And as I mentioned, within the African context, they're incredibly unique. You have obvious finds around the Persian Gulf, then towards India and Pakistan, and then down the east coast of Africa at sites such as Manda, Shanga, Kilwa, and Chibuwen. Importantly, most of the African dates for these finds are between the 8th and 10th centuries AD. That's Chibuwen down here. So if we're talking about the Letaba sort of river, it's way down there. And these blue-green glazed ceramics, with the dates that we have for them, are about 750 to 800 AD, right? So, which really puts it at the earliest phase of the exploration by traders down the East African coast. But more importantly, locally, it also coincides with a really steep increase in humans living along the Letaba and Olifants rivers, right? And so there's this correlation between the increase in trade and the first sort of evidence of trade, really, and the initial and this intensification of people living on the landscape, right? And the understanding is that we're arriving at is what's driving this increase in trade and population along the Letaba is the exploitation by these communities of the wild resources. The focus traditionally has always been on the exploitation of ivory and then later on gold, and in other words, just elephants in terms of the exploitation and the export of animal products to the Far East, right? But what we're showing at the Letaba sites is, especially during the initial phases of long-distance trade and the development of these connections between Southern Africa and the Far East, is that it's a wide range of animal products that are being sort of produced, consumed, and exported. It's skins, it is ivory, it is tusks, it is horns as well. And so, especially given the sort of recent sort of attention that rhino poaching, for example, has been gaining, and the export markets in the Far East, we're thinking it's a modern phenomena as a result of globalization. It is actually not, right? These animals have been exploited for at least 1,400, 1,500 years, and these products being exported towards the East Coast. And so the significance of these sites are that it really demonstrates the earliest phases of international trade into Southern Africa, South Africa becoming part of a global sort of community through trade networks 1,500 years ago, but also that the wild resources of regions such as Northeast South Africa are incredibly important, and that is really what's driving this initial sort of interest into the Southern African interior by outsiders. Right, thank you very much.
Thank you so much, Alexander. There's probably 1 minute, or maybe rather ask after the second talk, which will also be presented by Alexander, by any Antonitis hunting communities, animal resources and trade in KNP.
Right, so Annie couldn't make it, so I'm going to read her talk, but I literally received this during tea. So if I do stumble, I ask for grace. Anyways, right, so giraffes. It's what it says on the slide here, right? Okay. So as I mentioned, Kruger Park's been known for its wildlife, right? And the one thing we typically associate with Kruger is that it's unspoiled, pristine, and it's an area for natural heritage. However, the modern day absence of people on this landscape creates a misleading perception about the area's rich cultural heritage. People have lived and built up generations of ecological knowledge since the later Stone Age and possibly even earlier. Kruger Park wildlife is omnipresent today, as they were in the past, and this paper will talk about accessing data on these animals and how people interacted with them in the past through an archaeozoological approach. Archaeozoology is a well-established field in South Africa, and for those of you who aren't familiar with the term, it is the study of animal remains from archaeological sites and is specifically interested in the relationship between these animals and people in the past. This talk will cover faunal remains from the Iron Age sites I just mentioned in the previous talk, and during looking at the excavated material that we generated recently, as well as the older material that was excavated at these sites. During these initial phases, a PhD study was done by Ina Plug from then the Transvaal Museum, who illustrated a deep-time human-animal interaction in Kruger Park based on faunal material excavated by University of Pretoria. Just to provide some background to the methodology, archaeozoology uses comparative anatomy to identify animal species, skeletal elements, age, sex, and so forth from fragmented faunal remains. There are a few comparative skeletal collections in the country located at museum collections, but the De Tsong National Museum of Natural History, the old Transvaal Museum in Pretoria, has the only skeletal collection that caters specifically to archaeozoological research. And what you can see here is a lion's skull fragment being compared to a modern one, right? And through basically using these comparative collections, we're able to identify species on these sites. Right, so archaeological fauna material from Letaba is typically either well preserved, like this buffalo mandible you see over there, or highly fragmented, as you can see in the tray over there. Fragmented material has been exposed to the surface to changing environmental conditions, animal trampling over centuries. Such bones are very difficult to identify. Luckily, the Letaba material is fairly well preserved with enough distinguishing characteristics to present on the bones to identify the family, genus, or even species. The bones also preserve evidence of butchery marks and other cultural material modifications. That's Ina Plug's original notes. We're just going to skip over that. Now, back to Ina's findings. For alleys 6 and 7, she found that subsistence was based on big game hunting, supplemented by limited reliance on livestock. On the right is her taxonomic list for alley 6, which is the one side, which consists primarily of wild animal taxa, including buffalo, giraffe, and rhino. And this is a rhino humerus that was excavated in 2021. This minimal livestock presence led both Ina Plug and the excavator Andre Meyer to regard Iron Age settlements in Kruger as a socio-economic backwater, populated by ephemeral communities whose political development was constrained by tropical disease and climatic conditions that were unsuitable for intensive cattle herding, and these conditions forced people to move their villages on a frequent basis. Plug also recorded some human-animal modifications on the bones, but the data as reported here have been separated from the context that makes it meaningful. Of course, each study has its own focus, and Plug was interested in longer chronological and regional patterns. However, in thinking about targeted exploitation and butchery patterns, for example, this data table only has the number of bones that shows some butchery marks. The species and specific skeletal parts is unknown, which makes it difficult to understand how people processed the carcasses of different species and different sized animals. Questions about carcass butchery were addressed more than 25 years later after Plough's original study, when Grody looked at the data that was excavated earlier. She was initially interested in better understanding the large proportion of wild fauna at the site, specifically looking at the carcass processing and what it might reveal about the predominance of wild animals there. But she expanded the sample of identified specimens and found large numbers of buffalo, blue wildebeest, and zebra remains, and very few cattle and even smaller or fewer livestock remains, small livestock remains. Thus, the same pattern that Plough identified, Evan was able to confirm. Other game included black rhino, eland, kudu, lion, baboon, warthog, and bush pig. And what you can see here is a baboon mandible compared to a modern museum specimen, right? There's the museum specimen and the sort of archaeological specimen over there. Grody was able to conclude that there was a very specific focus, not just on wild animals, but on exploiting large and very large wild ungulates, specifically gregarious animals. There are also a few sub-adult animals in the sample, meaning that adult animals in particular were targeted. In addition to large amounts of meat, this pattern also speaks to the skill of the hunters, time and effort involved in killing large numbers of these animals, and the risk involved in tracking dangerous animals such as black rhino, hippo, and buffalo. Carcass processing that suggests quick butchery by highly skilled butchers. Here you can see some of the examples of butchery marks on the bones. Those are all sort of butchery marks sort of on these bones. Marrow was selectively extracted from long bones, and bones were not boiled to extract bone grease, specifically something that we see in other sites. But Grody's analysis thus suggests that carcasses were not fully utilized and that the selective or perhaps wasteful use of certain portions does not point to a situation where resource depletion or environmental stress existed. It does point to intentional choices and strategies, and in the case of the Letaba sites, she argued for specialist hunting and carcass processing on a recurring basis. However, it was not clear whether the extensive hunting was linked to long-distance provisioning of non-local communities or whether these animal products were being incorporated into local trade networks. New research conducted since 21 has provided an opportunity to revisit the reasons for extensive hunting of large game. This is especially exciting given the discovery of long-distance trade goods and the evidence of technology, of imported technology along the Letaba River. Here's some of the slides of animal bones being excavated. In one sort of 2-week excavation period, over 300 kilograms of bone was excavated, right? And just to put it in context, that's a lot. Preservation of the bone is incredibly good. As you can see, it's almost, a lot of it, the skeletal features are complete, or almost complete. Elements of buffalo, blue wildebeest, zebra, giraffe, and others. And there's also bush pig and black rhino, animals that no longer occur in those particular parts of the park. This photo sort of documents the recovery of a large piece of wildebeest skull, which is also a skeletal element that rarely survives in the archaeological record. Skull fragments of buffalo and waterbuck with the horns clearly chopped off were also found. This study, coupled with the data generated by Plough and Grody, will also provide useful data on previous ranges of certain species, which is important in understanding past resource availability and environments, as well as providing usable data for present-day management policies. In conclusion, the previous and ongoing study of animal remains from Letaba show that these Iron Age communities developed extensive knowledge of animal ecology that was transformed into tradable goods and sustained over a 400-year period. Importantly, Letaba hunting communities and wild animals were active contributors in the initial expansion of coastal interior trade networks. Thank you very much.
Thank you so much, Alexander. We have about 5 minutes. Any questions? Okay, here's Collie.
Thank you, Alexander, for those fascinating talks. I was just wondering, so you have all these large carcasses, and I assume that they were not killed right at the site because you soon, create some landscape of fear and chase animals off. So how did people bring them back to the site if they were such large animals, black rhino, for instance?
Yeah, so the whole concept of landscapes of fear, which I first encountered now during this conference, has been sort of eye-opening to me, because it's exactly the kind of question we've been struggling with, right? Is these large animals were clearly, I think, hunted at the site or really close to it, right? But how do you entice animals to come into where you're staying, right? So and I think there's, what could be important here is its location right next to the Letaba River, as well as a series of interconnected dongas, right? And it's incredibly deep dongas with about 12-meter high walls, right? And my suspicion is that potentially could be part of these pit traps that they're potentially using. And then with mass organized drives, potentially corralling these animals into these funnels that are created by these dongas and potentially killed that way. But this is absolutely speculation, right? I'm looking at sort of the topography of the landscape around it. This is what I'm getting at. Elephant we rarely find on these sites just because of that factor that you chuck off the meat that you want, you take out the ivory, and you leave the rest of the carcass behind, right? The fact that we're getting large buffalo and big pieces of the skeleton on the site as well, still articulating, means that it's killed really close to the site as well. So I've got some ideas about it, but how to test it is another matter, right? But it is taking place quite close to the site as well. That's what we know.
Hi, thank you for very nice presentations. The reference point in conservation is often the beginning of the 20th century. We always say that wildlife was that abundant, that diverse in the beginning of the 20th century, and we try to go back to that state. But there is an hypothesis supported by Livingstone's observation, especially along Lake Malawi in the 19th century, is that population in Southern Africa and probably in Eastern Africa have disappeared during the 19th centuries by the use of by the slave trade. And that could have opened a huge space for wildlife densities to rebound artificially, creating those wildlife densities at the beginning of the 20th century, challenging the reference point. Is your field investigating those kind of questions?
No, but we have data that can clearly talk to that, right? Because if you look at long-term trends extending beyond 100 years, right, this is where you start looking, right? Archaeology is the only data that really can give you this. And archaeological data is in a sense unedited as well, right? It's what people consumed is what they throw away, right? They don't really think about what they're throwing away. So it really does sort of provide us data. We're less able to talk about densities, for example, but variability and presence-absence data in terms of wildlife is obviously something that we'll be able to sort of provide. So the thing is the human presence on the landscape isn't stable either, right? Over time, there's a great variability of how people moved across the landscape. And obviously that has a knock-on effect in terms of their exploitation of the environment as well, right? So it's dynamic, as everything else. I mean, if the entire session yesterday was about how dynamic these savanna landscapes are, right? But we have to sort of factor the human presence into this dynamism as well, especially if we talk about long-term change. But yeah.
Hello? Yeah, hi. So I'm going to mangle this name, which is, I'm going to say Mapungubwe, but I know I'm saying it incorrectly. So there's evidence of East Asian trade at Mapungubwe. So is this contemporary or predating?
This is about 500 years earlier, right? So, yeah, just to put it in context, Mapungubwe is seen as the first kingdom, first sort of state-level society in Southern Africa. That's about 1200 AD, right? So and a lot of that initial political power is based on the existence of long-distance trade with the East Coast. We had never really gotten a grip on where this really starts, and it seems to start further south and much earlier than previously thought, right? Potentially here in the Kruger Park with these large rivers connecting the interior with the East Coast.
We can quickly squeeze in one last question from you, see?
Yeah, thank you very much. Very interesting results. I was wondering, I mean, this is now one site on the Letaba River. What do you think, how many similar sites were on the Letaba River and how many similar sites in Kruger Park?
Yeah, so we're focusing on one site. There's, as I mentioned, there's about 330 that were documented initially for the entirety of Kruger. Along the Letaba, there's about 60 dating to this period up and down the river, and there should be more next to, on the banks of the Olifants. The Olifants, however, because it doesn't have the steep terracing that the Letaba has, the preservation of sites are just not there, right? So we're missing what happened along the Olifants, but we have this for the Letaba. So the next phase of the project will obviously be to explore the other river systems as well. The Shingwitsi is one that's unexplored as well. But once again, it just highlights the importance of these river systems in terms of human movement, right? Because if you look at the earlier trade network, the early sort of European explorers and hunters and so on, everyone mentions that movement takes place next to the river, right? You don't have to carry water with you. It's a natural sort of incline. It's a path basically already there. And so these trade networks connecting the interior with the coast were all along these rivers. And so, yes, it's a very small sample size. These are by far the biggest early Iron Age sites in this part of the world, dating to this period. There's probably a few others of a similar size, but we're dealing with preservation issues. But as I said, this at least offers us some avenue into the past, right? And from here, we're expanding it out to other sites as well.
Thank you so much, Alexander. For further questions, you can continue the conversation over lunch or tea time. Thank you so much. Also to note, there's 4 posters associated with the session from Shante Banat, Tim Forsman, and Justin van Heerden. Right, moving along to the last session before lunch, we are at Wildlife and Health, and the first speaker, 15-minute talk, is from Gareth Hampson, Parasite Effects on Movement in Serengeti Wildebeest.
Okay, hello everyone. Fantastic to be back here, and thank you Sandparks for the opportunity to present here again. Okay, so today I'm going to be talking about parasites in wildebeest and the Serengeti ecosystem. And just to say up front, this work represents the efforts of many people. We're a big collaborative team. There we all are. And yeah, it's been fantastic to work with everyone. Jason is part of the team. He's going to be giving the next presentation, and that'll focus more on sort of the core focus of the project. What I am working on is collared animals, so looking at individuals through time. Okay, so with those individuals, I'm going to just have a look at these 3 questions today, just to see if there's any hint that parasites might matter to the wildebeest population, then to see where and when they might be getting infected by these parasites, and then to see if those parasites might have any effect on their movement through the ecosystem. Okay, so like I said, working on these collared individuals. There you can see a collar, and there's a second one. This is just a moment in time where animals collared years apart, cross paths. So my job is to go out and find these ladies, so out tracking the Serengeti, and then I collect their dung. So we're using the dung for a variety of purposes, so we take a range of samples. I'm only going to speak about the samples that we use for fecal egg counts today, so the egg counts or the parasites that are in the dung or in the animal. So those samples go to the lab. I've got a wonderful crew of people in the lab who have been generating these data. For the fecal egg counts, they use the McMaster technique. I'm only going to speak about strong gel nematodes today. Just a note on their life history. These are worms that live in the wildebeest gut. So the adults in the gut producing eggs that are dropped in the dung. They hatch into larvae, and the larvae's got 3 stages. And that 3rd stage crawls up the grass and then gets eaten by a wildebeest back into the gut. So something important about that is that with this free-living stage, there's an opportunity for animals to drop parasites into the landscape and then move away from them, sort of break their life cycle. Okay, this will give you a sense of the sampling I've been doing. So here are all the individuals I've worked on, and then this is sort of a timeline, and every time you see a little green start, that's when I got a sample from them, and then as it gets longer since I had a sample, then the color shifts yellow to orange. So I worked on 35 animals so far. There's 25 still going at the moment. Started in October 2022 here. Going to present data up to December last year. In total, we're on about 250 samples now, and we'll talk about 200 of those today. Typically sort of 6 to 10 samples per animal. I am making the most of my permit to roam around the Serengeti, so I've collected widely. So that's the distribution of those samples. And then you'll see in some analyses I refer to a dry season range and a wet season range. Dry season range up in the north of the system, and then the wet season down the southeast part, and then the rest I've just put as sort of a transitional zone. And just to note that the polygons I've drawn there are subjective just from my experience, so they can be analyzed formally, but just as a note. Okay, and so then when I'm with an animal, I also record some other data. So things like what their reproductive status is. Here's 103, there she's pregnant in January, with a little calf growing up through the year. So I record whether there's a calf there or not. Also record, give them a body condition score. So this is a visual score from 1 very poor up to doing pretty well. And then I'm also going to make use of some modus data in some of the analyses. And just to say, so those I'm using NDVI, and then a sort of time since fire variable that I've developed from modus products. Okay. Right, so to have a look at that first question, this is a quick and dirty analysis. I'm just looking at whether there's a relationship between an animal's body condition and its parasite burden, so that FEC, the fecal egg count. This is for all the wildebeest samples I've collected, so it's beyond the collared animals. And it's just, what we see is quite a clear negative relationship between body condition and parasite burden. So animals that are in poor body condition tend to have higher parasite burdens. It's not to say that the parasites necessarily cause that low body condition. I'm sure they do in many cases, but an animal might lose body condition for a different reason and then be more vulnerable to parasite infection. Body condition integrates a lot about your life and influences your recruitment and survival probabilities. So it seems very likely that parasites will be having effects that feed into population dynamics. And that is something that we'll explore separately. So there's just a quick note to suggest that parasites probably matter in the system. Okay, so now just have a look at where these wildebeest might be being infected by these parasites. So in this first little analysis, I'm just looking year-round, so my data points, this is the collared animals, looking at all the data I've collected. And so just fitting a negative binomial model, it's a mixed effects model, so I've got animal ID as a random effect just to account for the repeated sampling. And then I'm looking at the effect of having a calf or not on your parasite burden, and then which range you're in, so that dry, wet, or transitional range. And so what we see is that there's a significant effect here of calf presence. So you see that animals that have a calf tend to have higher parasite burdens than those without a calf. Interestingly, we also see that these parasite burdens tend to be higher in the wet season range than they are in the dry season range, and so fairly high in the transitional range too. Okay, so the next step then, I've just taken the samples that I've collected in the wet season range. So the variables in the model change a little bit. I've got now the amount of time that an animal has spent in the wet season range, the number of days in the wet season range, in addition to calf presence. And in this situation, what we see is that effect of having a calf falls away. So this comparison here is no longer significant up there. There's a marginally significant effect of a sort of a positive effect of time spent in the wet season range on your parasite burden. So it looks as though the longer you spend in the wet season range, your parasite burden probably increases. You can run the same analysis in the dry season range. And what we see here is that effect of calf presence is now very clear, quite distinct between animals that have a calf tend to have much higher parasite burdens. There's also a much more robust relationship between the amount of time spent in the dry season range and that sort of positive effect on parasite burdens in animals. And then there's also an interaction between those two terms. And sort of the main thing there is that it sort of seems these animals down here that don't have a calf, when they arrive in the dry season range, they tend to have very low parasite burdens. And then those sort of increase more steeply over the course of the dry season. Okay, so just to sort of pull this section together a bit, so we're seeing higher parasite burdens in the wet season range than the dry season range. There could be a number of reasons for that. I think one is probably to do with sort of microclimates. Or just the environmental conditions. It's wetter, that's more favorable to the parasites, the larval survival in the grass layer. So the larvae could be doing better. They might be more concentrated in a shorter grass layer in the wet season range, so more larvae per bite. Fire in the dry season range could be reducing parasite loads in the grass layer, or something else entirely. But those are the sorts of things that could be going on, creating that difference. The increase in time in parasite burdens over time in both ranges makes sense. I think there's not an opportunity for when animals are there, the parasites have got an opportunity to complete their life cycle, and so those burdens just sort of build up in the landscape. The whole parasite population increases in the landscape. With the calf effect, it's a big physiological toll, being pregnant, giving birth, and the whole lactation process. And I think through that, there's probably an element of immunosuppression that makes you more vulnerable to parasites, or the parasites in you do better. So I think that explains the difference. And the lack of effect in the wet season range, I think, is probably because most animals are pregnant, most adult females are pregnant when they arrive in the wet season range. So probably many of those without a calf have suffered neonate mortality. And my guess is that they've still gone through this big sort of physiological shock, and they haven't recovered from it yet. So I think that's what's going on there. But then by the time you get to the dry season range, say 3 or 4 months later, those animals without calves have now recovered, so they're doing a bit better. And then from that low base, they build up very more steeply through the dry season. Okay. So for the third question that I'm going to be having a look at, what I've done here is to take the GPS coordinates for each of the animals that I've worked on. So that's from their collars. The time steps aren't regular, so I've fitted a continuous time correlated random walk model, so I can get regular 4-hour intervals between locations. So some are predicted, some are the original data. And then I've fitted those in a hidden Markov model with the purpose of identifying 2 different movement states that the animals might be in. So those states are color-coded here. Let's start with the blue ones. And so this is sort of a traveling state. And the characteristics of this, so between successive GPS points, they tend to be further apart, so you're moving further. So that's that over there. And then your turning angle here is much narrower, so it's much more sort of directed, long-distance steps, as opposed to the orange state, which is sort of more exploring a space. You're taking shorter steps and with wider turning angles. So it's more like you're spending time in an area you may be foraging, but it's sort of a more sedentary phase. Okay, so going through all of that, I'm then taking these states and fitting a model now where I've got what's the proportion of time that an animal has spent in state 1, that more sedentary state, in the week either side of when I collected a dung sample. So I collect a dung sample. What portion of the time around then has an animal spent, say, in that foraging state? So a positive effect means more time doing that. And then I've looked at these predictors. So that fecal egg count, the parasite burden, calf, yes or no, NDVI, and then that time since fire variable. Animal ID as a random effect, and then also some attempt to control for the spatial autocorrelation, but just by fitting range as a random effect. Analyses on early phases, we'll tighten them up over time, but just to show you the patterns that are coming through. So what we see here is that all of these variables are significant predictors of the movement state of these wildebeest. In terms of the parasite burden, look, it's not a very strong effect. It's supported, but what you see is that animals with higher parasite burdens tend to move a bit more. So that's a negative effect there. If you've got a calf, and this sort of makes sense, if you have a calf, then you tend to be more in that state one, so more in that sort of foraging state. You're less mobile. Probably makes sense with the calf itself, just shorter legs. It's not, just spending more time foraging, not moving around as freely. In terms of NDVI, what we see is NDVI gets higher, you're more likely to spend time in that area, so less likely to be moving. So that sort of makes sense too. So perhaps if NDVI tells you something about foraging conditions, perhaps you're spending more time foraging in better areas. And then from that time since fire variable, a bigger number means more recent fire in that landscape. So we're seeing that animals are tending to move away from areas with more recent fire. I'm not entirely sure what the fire variable actually tells us about the landscape. It could be telling us about fire, but it could also be telling us, say, something about like how tall the grass is and how frequently it's going to burn. So there's a lot more digging to be done to understand the role of fire here properly. But suffice to say, whatever this variable represents, it's got some sort of effect on how animals move in the landscape. So for the last little bit now, then I've looked at the interaction between parasite burden and each of these variables. And so what we see here, again, this marginally significant effect. Animals that have a calf there, if they've got a high parasite burden, they tend to be more likely to want to move. Animals, okay, if you just look at the blue line, these are sort of the foraging conditions, the NDVI areas that they don't want to be in. The higher their parasite burden, the more likely they are to move out of that space. Or to show movement behaviors, yes, that shift in movement behaviors, and then the same with this time since fire. The black line is the more recent fires, landscapes, which is where they didn't want to be, and as your parasite burning gets higher, you tend to be more inclined to shift into a traveling sort of state. Okay, so just to wrap up then, so it does look as though these parasite burdens are having an influence on how these wildebeest move through the ecosystem. If their parasite loads are higher, they tend to want to move, so there might be this idea of just trying to break that parasite life cycle. There's a lot of theory around migratory escape, which we'll dig into over time. These animals with calves that want to be sedentary, they might be pushed by parasites to actually start moving, getting into say, cleaner areas. And then areas that have undesirable NDVI or these fire characteristics, if you've got a higher parasite burden, you're more inclined to try and get out of those areas. All right, so I think parasites matter in the system, and it's fascinating working on this new topic for me. All right, thanks everyone.
Thank you so much, Gareth. Unfortunately, you used all your time. Moving along to Jason Donaldson, the role and effects in resident parasite dynamics.
Yeah, hi everybody. Thanks for coming through. So I'm going to be discussing a similar system to what Gareth's talked about here. But really focusing on the effect of these migrants that are moving through the system on resident animals within local areas. And Yeah, so I'll just highlight the fact that what Garrett's been talking about within the migrants is something that's fascinated researchers of disease ecology for quite a long time. You have these super high densities of animals moving long distances, and you would expect that they have some sort of impact on disease ecology when you have such hypersocial animals moving these large distances. And so because of that, there's been a lot of research that has focused on the effects of migration on disease within the migrants themselves. So some things that we know is the fact that with migrants, they move throughout these broad areas, there's increased sampling of the landscape, and so they often have higher diversity of parasites within them. But then there's also these processes of migratory culling and migratory escape, which means as you move as a migrant, you might be escaping from areas with high parasite densities, but you also may cull animals in your population that have high infection rates, and so that can affect the likelihood that infection stays within the population. What we know far less about is about residents. So this is a tiny little topee out in this big vast herd of wildebeest, and the question is what's happening with them, and no one has really cared about that for quite a while. And the idea, when people have looked at this, has been that when you have a population of migrants moving through a population of residents, then some infection load is passed on to those residents as the migrants pass through. And this is defined as a transport effect, right? And so to get a bit more detail about what transport effects are and how they would work, I'm going to focus on a model system of gastrointestinal nematodes, which Gareth has spoken a little bit about. But essentially, within your host, you have your adult stage nematodes. They then lay eggs, which come out in the feces of these wildlife or the host species. The eggs of these hatch. L3 or larvae, free-living larvae stages, climb onto the grass, and then the host eats it and gets infected again. So this is the system that I'm going to be talking about today for the purposes of this talk. And if you imagine again that we have these resident animals, what transport effects do is it means that when your migrants come through, the wildebeest in this situation could add a lot of dung to the system. That dung would mean more eggs, more fecal, more larval load, and so when the migrants leave, the risk of infection for your resident animals is then much higher. Okay, so this is the idea that migrants are transporting infection load into resident home ranges. But there's a second thing that migrations do when they get to this sort of scale. And if you look at this picture, it's fairly obvious that these animals are using a lot of resources. They need to feed, they need to drink water, and so they're going to have trophic effects on your resident animals wherever they go. And that's something that we've been very interested in, and has largely been ignored in your kind of traditional understanding of what migrations are doing to parasites within resident animals. And so we recently argued that migration should not only have these transport effects, but they should also have very extreme trophic effects. And why we're arguing that is essentially, again, to use wildebeest as an example here, when you look in the Serengeti ecosystem, what you can see here is these wildebeest are grazing this grass really short, and you have this tall grass here as well. So there's a lot of feeding going on by huge densities of animals. What is that doing to things like your parasitic larvae that are in that grass layer? The first effect that could be happening is you could have this direct impact, right? That's just the consumption of the wildebeest. So again, we have this resident population with some parasite load in this grass layer. Your migrants come in and they eat that grass layer. What are they doing? Well, they're consuming these free-living larvae as they feed. So they could eat those free-living larvae and then they could leave the system. And when they leave, they may leave it cleaner than when they arrived, right? So there's this capacity for a direct removal. But again, there's a third potential mechanism here, and that's indirect effects, so vegetation-mediated effects. So again, you see these wildebeest are grazing this grass really short. What impact does that have on larval survival? So from work we did last year, we looked at the effects of fire and parasites, and what we found there was this really strong impact of grass height. So here we have bin grass biomass on the x-axis, soil surface temperature on the y-axis. And what you're seeing here is as you get an increase in grass biomass, you get a cooling effect on the ground temperature, right? When we compare that to the survival of parasites in these plots that we have, we see that here on the y-axis we have change in parasite larvae per kilogram of dry matter of grass. And on the x-axis, we have this ground temperature, and you see this decline in survival. So you're getting a loss of larvae when there's high temperatures at the ground level. So if we think about that in the terms of the system, inside this tall grass area, you're going to have cooler temperatures. Outside, you may have hotter temperatures. And so the wildebeest may be having this environmental impact that's killing larvae, right? So that's this indirect effect that we talk about. So we were interested in these questions. We wanted to know what is the actual impact of migrations, of the wildebeest migration when it moves through resident home ranges. And we used the Serengeti ecosystem as a model system. It contains about 1.3 million wildebeest that move throughout the area. And they interact with a myriad number of resident animals. And these residents are far more locally constrained. They don't travel throughout the entire system. And so, as I've mentioned, we thought that the wildebeest would have these transport effects, and then they should have these trophic effects. And so that's what we wanted to explore. To address this, we've been working with a really big group of researchers from the University of Glasgow, University of Georgia, Yale University, Wake Forest University, and the Tanzanian Center for Research Cooperation. And we've been exploring these questions for about the last 2.5 to 3 years. So the first question we're interested in is, do wildebeest exert these transport effects? We wanted to test that first. We then wanted to look at whether they had trophic impacts on parasite transmission in residents. And importantly, we wanted to distinguish what those trophic effects were. What was the mechanisms behind the trophic impact? And so we've been looking at resident animals here. This is the snapshot Serengeti grid, and you can see we've had this focal region that we've been looking at resident herbivores within. And we've been collecting dung samples. So we've been going out and collecting dung samples every month of these 4 focal species to try and get an idea of what the fecal egg counts in those dung are. We've then had our tame roaming wildebeest in the form of Gareth, who's been moving out throughout the whole system, and you can just see the spread of points that he's been collecting. Again, collecting fecal egg counts data for the wildebeest population. So we know what the migrants are doing, and we can then work out what impact they're having on the residents. So this is just some data to show you sort of the background of these infection loads for residents versus migrants. What you can see here is that Grant's gazelle have by far the highest gastrointestinal nematode burdens of all species. Sorry, on the y-axis here is gastrointestinal nematode eggs per gram of fecal matter. And then this is just time through our entire project. And so you can see consistently high impacts or infection loads in gazelle, much lower in your migratory wildebeest and your buffalo. Okay, so what do our results look like? So this is a comparison for the 4 resident species. So we've got hartebeest, topee, Grant's gazelle, and buffalo. And again, strongyle eggs per gram within the fecal matter on the y-axis. On the x-axis here is wildebeest dung per meter squared, and this was taken from fixed plots that we monitor each month within our resident home ranges. So we can link the dung of our animals where we're collecting the fecal egg counts to what's going on in the pasture nearby. And what you can see here is there's quite a few different results going on, but there's definitely responses of resident animals to the presence of wildebeest dung in the system. The thing I want to highlight is that for every single one of these species, there was a season by wildebeest dung interaction. So the season that these interactions are occurring really matter to what the directionality and impact of wildebeest is on resident infection loads. What I mean by that is that if you look at the color of these lines, the blue lines are from the short rain and long rain seasons, and the red lines are from your dry seasons, right? And what becomes fairly clear is that we only see transport effects during the wet season. So it can be the short rains or the long rains, but we only see transport effects, that bringing in of infection loads and increasing the risk for residents in the wet season. We don't see this in the dry season. Comparatively, we see trophic effects occurring in the dry season mostly. There are indications that it can happen in the wet season in Grant's gazelle and hartebeest, but the majority of the effects we're seeing are happening in the dry season, which raises the question, why is that occurring, right? And that gets to exploring these mechanisms. So The first thing we did to explore these mechanisms, and this work was done by Hussein Kamara, who's a PhD at University of Glasgow, was we had these experimental plots. At day 0, he added a whole bunch of dung that had a known amount of known fecal egg count to both sets of plots. And then at day 14, which is when these larvae have emerged and are reaching their peak abundance in the grass layer, he clipped grass to 5 centimeters to represent a wildebeest migration coming through, grazing that grass short. He took that grass pasture and he measured how many larvae were in that grass pasture. And then he took separate samples to look at what the resident exposure was in these controls versus these treatments of wildebeest migration. What you see here is when wildebeests come through and consume, they're eating a large amount of the larvae, which are sitting high up in that grass layer. That leaves far less exposure for your residents. And ultimately, that means that resident exposure is much higher when there's no migration versus when there is a migration. To explore the seasonal impacts, we had these plots where we mowed the grass very short, and we added dung to these plots again. And on day 0, we looked at what was in the grass layer. Day 14, you see that change, and then day 21. This was during the dry season, and what you can see here clearly is that on the treatment where we clipped it, there was less survival on those plots versus in the tall grass. So grass cropping in the dry season means there's more gastrointestinal nematode mortality in the grass layer, lower infection risk, because they're dying from that hot, dry conditions on the short grass. Comparatively, in the wet season, we did the same experiment, and you can see that it switches, completely changes here, and so you get higher survival on the cropped grass versus the tall grass. So what's happening here? Well, what we think is happening is the conditions are ideal for emergence and survival of these larvae, but it's on a shorter patch of grass, and so higher concentration per dry matter herbage. And so you're concentrating this. So ultimately what this means is that you have these two distinct phases of migration impact. Transport effects do not occur in the dry season because the larvae die. They do occur in the wet season when there's good survival of the larvae. The direct trophic effects occur both dry and wet season, and both reduce the impact of, or reduce the risk of infection for residents. And indirect trophic effects switch between seasons. So in the wet season, you get this increase in risk, and in the dry season, you get a decrease in risk for your residents. And so if you couple this all together, you can start to understand what's going on with all these different interactions at different points in time. We do still have a lot of questions. I think one of the really fascinating things, and this goes back to the great session yesterday on trophic ecology, is to say, why are these different species reacting differently? What are the interactions going on between the species that means that one set of them is getting impacted by migrants when the other is not, and the direction can even be different? That must have something to do with their resource sharing, the niche partitioning that's going on, and I think that's something that's really still wide open. And so we're kind of addressing those things now. Yeah, so thank you very much to TANAPA and to WIRI who supported the research, and to the funders. Thank you.
Thank you so much, Jason, for the talk. Unfortunately, we have run out of time. And right now, we'll be having Lizel Retief. We'll be having Lizel Retief, rodent-associated hemoplasmas, assessing disease dynamics at the wildlife, livestock, and human interface.
About hemoparasites and their dynamics in a protected area and surrounding areas. Oh, sure. Yes, I've got a very soft voice, sorry about that. So at the intersection between protected areas and human settlements, a dynamic environment is created where pathogens can move bidirectionally between wildlife, livestock, as well as humans. And within these environments, rodents are quite important, and the reason for this, there's quite a few reasons for this. Rodents are the most widespread and diverse order of mammals. They're also very good at forming close associations with human settlements, and they are vectors of various, or reservoirs of various pathogens. So studying disease dynamics in rodents at this interface becomes quite important. And a group of bacterial pathogens that have recently garnered interest worldwide are hemotropic mycoplasmas, or hemoplasmas. Now, hemoplasmas primarily infect the red blood cells and can cause anemia and can lead to fatalities in a range of different mammalian species, including domestic species, synanthropic species, including various rodents, as well as other wildlife species. And recently it's been shown that it may cause quite severe infections in humans as well, with some fatalities recorded. So really understanding the dynamics of these pathogens are quite important. This is a review that was published in 2020 that looked at hemoplasma research that's been done in free-living species. And as you can see, especially for the order Rodentia, most of the research has been concentrated in specific areas, primarily in South America. So not a lot is known about hemoplasma dynamics, especially in Africa. So since then, we have looked at hemoplasmas in rodents. We looked at hemoplasmas in mole rat species and also invasive ratus species within Gauteng. But there aren't any data available really trying to understand hemoplasma dynamics in a range of different species sampled at the same time, also sampled within this, at this interface between wildlife and domestic species. So this is quite important because in terms of hemoplasma research, not a lot is known. It's currently debated whether or not hemoplasmas are transmitted through ectoparasites, or if hemoplasmas are transmitted through a direct transmission route, such as when species have aggressive interactions between each other or through vertical transmission. Furthermore, it has been suggested that there may be some barriers that prevent specific hemoplasma genotypes from moving from invasive rodent species like ratus to indigenous species, and it's not really known whether or not this is the case. In addition to this, there's not been a lot of studies that have looked at hemoplasma transmission between protected areas and more transformed areas. There's only one study that has looked at this in raccoons, and they found a higher hemoplasma prevalence in raccoons sampled in a protected area compared to raccoons sampled in a more urbanized area. But not a lot is known about what the trends are in terms of rodent species. So that brings me on to our research study. We did research within the Manisi community area, which is just a little bit north of where we are now. It consists of the Manyeleti Nature Reserve and then also villages within the Bushbuck Ridge municipality. And we collected 8 different rodent species within these environments. I've only shown 7 species on the screen because Mastomus natalensis and Mastomus cucha are cryptic rodent species, so you can only distinguish them based on genetic characterization. So we collected these species and then we screened them for hemoplasma prevalence. We screened lung samples to get an estimate of the occurrence in these rodent species, and then we also wanted to look at transmission routes. So we screened mouth swabs as well as ectoparasites that we found on the rodent species, and then for the samples that we did have available, we screened fetal samples of hemoplasma positive females. And then we also assessed whether or not there was a difference in hemoplasma occurrence between different habitats, so peri-urban versus natural, as well as sampling season, host species, host sex, and then individual ectoparasite load. And onto our results. We found no difference between the peri-urban environment and the natural setting, but we did find quite a high occurrence of hemoplasmas within this environment, as you can see. But where it gets interesting, the reason we thought that this may be the case is because we sampled right on the boundary of Manyeleti Nature Reserve, and this is a picture that I took standing inside the park right at our sampling site. And as you can see, we're right on the border of the reserve, and the villages where we sampled is right outside. So potentially what's happening is that we're not sampling at a fine enough scale, and we may not be seeing the differences between this protected area and more transformed areas. But where it gets interesting is when we look at the hemoplasma diversity. So of the 8 species that we sampled, we managed to detect hemoplasmas in 6 different rodent species, and we got quite a high diversity of hemoplasmas within these rodents, detecting 8 novel hemoplasma genotypes. This is our phylogenetic tree, and when you look at the genotypes that we detected, they cluster within 4 different well-supported clades. And I briefly just want to go through these different clades. So in terms of the invasive species that we sampled, we did detect only 1 hemoplasma strain, and this strain clustered well with strains that have previously been detected in rattus species at different parts of the world. And this supports the theory that these hemoplasma genotypes or strains are likely specific to the rattus genus and were introduced to South Africa during historic introductions of these invasive rattus species. When we look then at the hemoplasmas that we found within the indigenous rodents that we sampled, we can see that we had quite a one group of genotypes being found in all the sites that we sampled, and again only found in indigenous species. And when you look at the genotypes that we find, genotypes were either genus-specific or species-specific. The same goes for the other genotype that we detected in Ethymus ineptus and another indigenous species within this environment. Again, this clustered well with hemoplasmas previously detected elsewhere, but seemed to be species-specific. However, for the last group of hemoplasma genotypes that we detected, they clustered well with hemoplasmas previously detected in Rattus novegicus from Hungary. And what this shows is that while it's likely quite rare, historic host switching events may have occurred. And this shows that by introducing these invasive rattus species within these environments, we may be changing the pathogens that are cycling within our indigenous rodent species as well. And that's why also looking at transmission routes is quite important. So when we looked at the different ways in which these pathogens may be moving between different rodent species and within different species, we found that there was no mouth swabs that tested positive for hemoplasmas. And this is similar to our previous study where we looked at rattus samples, where we were only able to detect 1 hemoplasma positive sample when we screened 99 rattus samples. And this is likely because hemoplasmas can be quite, have an acute phase as well as a more chronic phase, and hemoplasmas may be transmitted through direct transmission during the acute phase when bacterial load is high, but this is likely not the main route of transmission. However, when we look at ectoparasites, we found quite a high prevalence, and this also was further supported by the fact that there was a significantly higher hemoplasma prevalence detected when there was a high load of fleas and lice also found on the rodents that we sampled. And then very interestingly, we found that 30% of the fetal samples that we screened were also positive for hemoplasmas, which suggests that vertical transmission may be quite an important mode for how these pathogens are moving from one individual to another within these environments. So then just to conclude, within the Menisi area, there is quite a high occurrence rate of hemoplasmas. And that it's clear that within this environment, what's likely occurring is that these pathogens are being transmitted through ectoparasites, but we also need to consider vertical transmission. And then finally, while there may be some species barriers that exist in hemoplasmas moving from invasive species to more indigenous species, historic host shifts may occur and need to be considered in future. So with that, I just wanted to thank you and thank everybody that has worked with me. And yeah, I welcome any questions.
Thank you, Lisa. We have time for one quick question. Anyone? Colleen.
Thanks for your talk. I'm not so clued up with animal health, but I assume the big challenge is rodents moving around and, like you say, infecting other animals like livestock. And if this happens from wild animals to domestic animals, I mean, is that possible? And then I was also wondering if something like Mycoplasma bovis, can that also affect other animals, or is it very specific to cattle?
I'm not sure about Mycoplasma bovis. The reason why is that hemotropic mycoplasmas, although they're currently classified within Mycoplasma genus, they're completely separate. But it's definitely possible that there are different diseases that can move from rodent species to other animals. An example is encephalomyocarditis, that is Mastomys species, the species that I spoke about that's cryptic. Those two species are known reservoirs of this virus. And when there's population explosions and of these rodents, because they're opportunistic breeders, they may shed these viruses into the environment, and it led to mass elephant die-offs during 1999. So it's definitely possible that these different pathogens can move to different animals as well, which is why studying and really understanding rodent pathogens within these environments is quite important, because they carry a range of different pathogens.
Okay, thank you so much, Lizel. Moving right along, we have Virginie Rugeron. I hope I'm pronouncing right. Xenosurveillance, a non-invasive, multifaceted tool for tracking zoonotic pathogens and monitoring wildlife biodiversity in real time.
Thank you. So I'm a researcher at CNRS in France, but I'm research associate since 2 years in Nelson Mandela University in Georgia. And today I'm here to talk about 2 studies that we performed using a non-invasive solution to screen pathogens in wild animals using xenosurveillance. So it's well established today that about 60% of emerging infectious diseases in humans are of zoonotic origin, meaning that they are the result of the transfer of pathogens from wild and domestic animals to human populations. Today, despite the advances in research for a lot of infectious disease, we still don't know what are the animal reservoirs, and we still struggle to understand the dynamic of certain infectious disease. This is mostly due to the fact that we struggle to collect samples on wild animals, which is highlighting the necessity to develop new tools to detect and track pathogens in wildlife and domestic animals over time. And that's not an easy task, especially when you work on blood-borne pathogens. I'm sorry, I'm sick, so I'm losing my voice. Huh? So traditionally, when you work on blood-borne pathogens, you used to collect samples directly on bushmeat. However, you never know really if the samples you are collecting is really fresh, which is associated to the conservation of nucleic acids. Also, you never really know where the animal got killed, so its origin. And as it's also part of the human diet, some of the time we struggle to get all the permits to collect such samples. Then we can collect these samples on carcasses, so this time we know the origin, however it's quite difficult to find. And it's also sometimes quite dangerous because the animal could have just been died from an infectious disease. So the last option we have is capturing directly the animal, but it's very expensive and that could be quite dangerous for humans and animals. So we are still facing challenging in obtaining a lot of animal-wide samples that are very diverse and that we could collect over a long period of time. One solution that could be used then is xenosurveillance. That changed completely the presentation. I don't know what happened, but it's weird, but that's okay. So we will use xenosurveillance, that is a tool to monitor pathogens in hematophagous organisms, so that could be insects, worms, etc. And the goal is to gain a better insight into which animals are the reservoir of the infectious disease, what is the dynamic of the infectious disease, and potentially we can highlight some health risks. Methodology talking is quite easy to use. You just have to collect the hematophagous organism and then screen them for the presence of the pathogen. In our case, we decided to work specifically on hematophagous flies. We chose these insects because they are very easy to capture. Both males and females engorge themselves, as the opposite, for instance, to mosquito females. We can get very important blood meal, up to 500 microliters. And also, they are known to feed on a very large diversity of vertebrate species. So we use hematophagous flies to explore the diversity of malaria agents. So malaria agents are due to a protozoa parasite, Plasmodium, that is characterized by a digenetic cycle with a part in a mosquito and then a part in a vertebrate host in which there is a large part that is happening in the red blood cell. As we can see here on this phylogeny, that is representing only a small diversity of the Plasmodium species. So we have a large diversity of Plasmodium species able to infect a large diversity of vertebrate hosts. So the goal of our study here was to investigate the possibility of using hematophagous fly to explore the diversity of these parasites in wild vertebrates. So to do that, we went in Central Africa, in Gabon, and we collected these samples, these flies, in 4 different parks, highlighted here in this map with a red dot. We collected those flies with traps that we called Vavois traps. That's traps that we do homemade. They are done of blue and black fabrics that are attracting the flies. Then once the flies is on the tissue, on the fabric, they are going up to this net cone, and they end in this little net pocket at the top from which they can go outside. So every morning we are putting the 25 traps in the site, and every late afternoon we are collecting the traps with the flies. We collected 1 month in each of these different sites, and we ended with a total of 4,099 hematophagous flies. So every day on the field, when we were getting back to the lab, to the field lab, we were dessecting each flies to collect the blood meal they had, and then we were putting their blood meal in tubes containing RNA later, which is allowing us to conserve the nucleic acid stability. Then once back to the lab, we extracted the DNA of each sample, and we screened specifically to diagnose the presence of Plasmodium parasites on a PCR based on the cytochrome gene. On the same samples, we also looked at the identification on which vertebrate host species the insect fed, using the cytochrome oxidase 1 gene. So then we analyzed all the results. I don't go into details. Sorry. And then what we've been able to show is that we first are able to identify and to detect a large diversity of vertebrate species, going from different herbivorous species, but also crocodiles, snake, bird, great apes, etc. So that could be a quite interesting tool to look at the diversity of vertebrate species in a place. Then we screened for the presence of Plasmodium parasites in each of these samples. And we've been able to show that we've been able to identify Plasmodium parasite infecting non-human primates, but also tortoise, birds, and antelopes. And that's the first time that we identified, by the way, species being able to infect antelopes, suggesting that's also a tool that could allow you to detect new pathogens in wild animals. So this study allowed us to validate the use of hematophagous fly to detect and track pathogen in wild animals. And we wanted to go a bit deeper in this study, and we decided this time to screen not only one pathogen, but a panel of different pathogens. This time, we went to Tanzania in the Ruaha National Park. So we went there because it was a park characterized by a high diversity of flies and also a large diversity of vertebrate species. It was also known for issues of spillover between wild and domestic fauna, with cases of brucellosis, Rift Valley fever, and bovine tuberculosis described. This time we went to this park with the same traps, but we only had 14. We put those different traps in 45 different sites, and we also ended to put a trap on the back of the car, because surprisingly we're collecting a lot of flies at the back of the car, for whatever reason, I don't know. And during this field mission, we only collected during 6 days. And after 6 days, we ended with more than 3,000 hematophagous flies. So we're happy, but we didn't took enough tubes to put these flies. So that was quite a difficult situation, but still we dissect individually each flies, and we decided to pull the flies, the blood meal of the flies, in tubes, again containing RNA later for the conservation of nucleic acids. Then, once back to the lab, we extracted the DNA of those pools, and we screened those DNA for a panel of different pathogens. So for 4 different parasite species, Plasmodium, Theileria, Babesia, and Leishmania. For 2 bacteria genus, I think it's Brucella and Mycobacterium, sorry for the mistake. And then for 6 different viruses. And then we also screened those DNA in this pooled tube for the content in the blood meal, so to identify on which vertebrate species each fly got fed, sorry. So for the results, as you can see here again, so that the pools in the abscess and the diversity of vertebrate were identified, we've been able again to identify quite a large diversity of vertebrate species, and in 6 days it corresponded to 25% of the vertebrate species described in this park, which is again suggesting that it could be a quite interesting tool to look at the biodiversity of vertebrate species in a park. So then for the parasites, so we screened all those pools for the parasites. We didn't find any pool positive to Leishmania and Babesia, but we found 1% of the pools positive to Plasmodiums, and these pools were containing blood from lesser and greater kudus. Then we found 5% of the pools positive to Theileria, and those pools were containing blood of herbivorous species going from lesser and greater kudu, but also warthog, giraffe, buffaloes, elephants. Then regarding the bacteria. So we identified 1% of positive pools to Mycobacterium, and those pools were containing different herbivorous species again, so warthog, kudu, giraffe, elephant, and buffalo. And 2% of the pools were then positive to Brucella bacteria. And those pools were containing blood from giraffe, from kudu, and from warthog. Then we looked for the screening for viruses, and we were quite disappointed because we did not find any positive sample to any of the 6 viruses we were screening. So we're like, okay, that could be due just to the fact that most of the time viruses have outbreaks in the time, so waving dynamics, so maybe we just collected in 6 days in a period where there was no viruses circulating. So we thought, okay, let's still continue to dig and find something to do with these samples. So we thought, okay, let's track for antibody that could be an indicator of the exposition to an infectious disease. So that's what we do, and we decided to work specifically on the pools for which we had a higher number of one species, which was the warthog pools. So we screened all those pools for the presence of a virus that we also knew was circulating in this park. So that was for one viral disease named the African swine fever virus. So to do so, we simply use an indirect ELISA that was targeting 3 different antigens, and we ended with 21% of seroprevalence in our study. So this highlighted that we could both use hematophagous fly to screen for molecularly for parasites and virus and bacteria, but we can also use it for the screening of antibodies. I'm sorry. So for the take-home messages, hematophagous flies look like a quite promising non-invasive tool to monitor blood-borne pathogens in wild animals, but also, why not, in domestic animals. It's also useful to look for antibodies as indicator of exposition to a specific infectious disease. And in more extent, that could also be useful to monitor the biodiversity of vertebrate species in a specific place. Finally, I just want to highlight that pooling samples is sometimes not the best way to look at specific animal reservoir that are infected with different infectious disease. And depending on the question you have to maybe think of bringing more tubes on the field. So I'd like to thank all the institutions who allowed this work and the fundings, but also the students, the postdoc, researchers and technicians and guides who allowed this work on the field. Thank you for your attention.
Thank you, Virginie. I think we have some few minutes for a question or two, just quick ones. Any question?
Okay, shout. Sorry? No, we didn't screen for trypanosoma. We didn't screen for trypanosoma, but we could have. Indeed.
Okay. Anyone? Gone. Thank you so much. Next speaker is Alexander Caron, and African Savannas and the One Health concept.
Thank you. Good morning, everyone. My name is Alexandre Caron. I'm a French researcher working for CIRAD and currently based at the Eduardo Mondlane University in Mozambique. I'll be giving this talk today on behalf of a lot of people, but I'll come back to that later. And I would like to discuss today how we can protect African savannas with the use of the One Health concept. So I'd like to come back to the conclusion of my talk last year, where I say first that the future of African savannas lies mainly outside protected areas for different reasons. First, that most savannas exist outside protected areas. And secondly, as Alexander showed this morning, we know now that pastoralists and their livestock have been keystone species of these savannas and engineering them and maintaining them for millennia. So I think it can be quite interesting to conserve savannas with human inside as well. So if you want to conserve savannas outside of protected areas, you need to look at the international agreement. The first one is the COP15, that we need to protect 30% of the earth by 2030. And we're currently at 17%, I think, in Africa. And the other one is the SDG goal that we need to aim at zero hunger by 2030. So if we keep on going with the land sparing approach that we have for conservation currently, we need to find more land for conservation, and that will clash directly with the need for more land for agriculture, because population in Africa is going to double before 2050. So my second conclusion was we need to co-design and try to promote a more land sharing approach, and so try to co-design shared landscapes, shared savannas, with different types of objectives. And if you want to do that, I emphasize that environmental justice is very important, and it needs to transform the relationship that we have between people and nature. Environmental justice say that sharing benefits of conservation with local stakeholders is important, but it's not at all sufficient. You need as well to work on to involve local communities and local stakeholders in the governance system, in the decision making about their own livelihoods, but also about protected areas, and that those people have huge indigenous knowledge systems, local culture that can and that need to contribute to how we govern those protected areas and their surroundings. So let me come back to the two entities I would like to mention here today. The first one is a research platform called Production and Conservation in Partnership, which started in 2007. Today, it's a large platform of universities from Zimbabwe, from Botswana, from Zambia, from Mozambique, and from South Africa. And also to mention the Zone Atelier Wange, which is a long-term social ecological research site that has been going on for more than 20, 25 years. And our common objective is to try to go towards a self-sustaining and functional social ecological system for the betterment of life of people and animals within Southern African protected areas. So if we think land sharing and shared savannas, we want them to achieve biodiversity conservation objectives, and this asks the question, what type of biodiversities? Large mammal, insects, other type of biodiversity, and also local development and well-being objectives. And we want savannas as ecosystem contributing to people, and also people contributing to savannas as in the spirit of nature contribution to people. So how can the One Health concept help on that? The One Health concept is driven by a quadripartite association of FAO, the World Organization for Animal Health, WHO, and UNEP. And they have a kind of a small IPCC group of experts called the One Health High-Level Expert Panel. And 2 years ago, they came up with a new definition of One Health, which is much broader from what you have heard before, that it is a concept to try to link more human health and animal health. This is much broader. On the left-hand side here, you can see that it promotes intersectorality and interdisciplinarity between the environment sector, the animal sector, and the human sector. And that needs to be done at scale, from the local scale to the national scale and even international and global scale, with important aspect of inclusivity, equity, and equal access as well. So if you want to capacitate those professionals to work together, you need to capacitate them in soft skills: communication, collaboration, coordination, and capacity building, so they can deliver One Health, which is now healthy ecosystems with healthy animals, healthy humans, and healthy plants. And I would like to just stop 1 minute on the concept of health. If you look at the definition of WHO for human health, it has two components. There is the biomedical component. It's about the absence of disease and trying to eradicate disease from your human population. But the second component is more holistic, and it's talking about mental health and well-being. And I think some presentation this morning touched a bit on that. And that concept, that definition of human health can be applied to animal health, animal disease, animal welfare, we heard that this morning as well, wildlife health as well, and also other type of health. So 3 years ago, we published a paper entitled Health in and Health of Social Ecological Systems, a practical framework for the management of healthy savanna in your case. And now I'm going to try to explain what we mean by health in savannas and health of savannas. So if we talk about health in savannas, it means that we need to deal with the different components of health within savanna ecosystems. It means public health, it means livestock health and welfare, it means wildlife health, it means rangeland health, it means also crop health, it means soil health, and let me just, you know now that more than 55% of biodiversity are in the soil. And we don't have a clue if our network of protected areas today is protecting that biodiversity. And probably that shared savannas and agro-system may contribute to protecting that biodiversity. Water health, we had freshwater presentation as well, and of course human well-being. So those are all the different health components that can help to address conflicts and opportunities within savannas and try to have those shared savannas. And I will only cite that report from the EU Eclipse request on biodiversity and pandemics that I contributed to, and just 3 of the recommendations here. If we want to work on those health components within savannas, we need to promote the emergence of a science of the wildlife-livestock-human interface. This is about disease transmission, but not only human-wildlife conflicts and all these kind of things that you can find in savannas, especially outside protected areas. And if you want to do so, you need to integrate much more social sciences and humanities, because all those conflicts are about conflicts between people, and less between nature and people, and animal and people. And if you want to do so, of course, you need to include much more local communities within the science of the interface. So now we're switching to the health of savannas. So what are healthy savannas? Here you need to borrow from the ecosystem health domain, and from Ostrom also, the social ecological system framework, where you have a coexistence of people and different types of natural resources within the same ecosystem. And the most important part is about system thinking. And this goes back to the intersectoriality and interdisciplinarity I was mentioning before. It means not only, we've been trained in very sectorial, I'm a vet, I've been trained in doing vet science, but we need to be able to work with grass ecologists, with agronomists, with social scientists in order to achieve that. And so, in order to have a more holistic approach of the system, very complex system, and so use also a lot more participatory sciences and the professionals that are trained in that domain in order to help us. So the health of those savannas or shared savannas need also to be connected with protected areas. This is a slide from last year. And in order for our protected areas not to be highland of conservation, they need to be in interaction, provide services to outside, and not only these services, and trying to, in a second step, maybe receive services from the outside surrounding lands. So it may seem very theoretical and complex, and it is complex. It takes a lot of time, it takes a lot of interdisciplinarity, it takes a lot of money, but we've been doing that for the past 7-8 years through different projects across the region, trying to engage local stakeholders and asking them, how can we improve your livelihoods within those savannas outside protected areas, within transfrontier conservation areas. And we co-design with local stakeholders intervention around natural resource management that they have selected, and mainly around water, crops, rangeland management, rangeland health. And of course, the assumption is that by creating more empowerment, more ownership of those interventions, we can reach more sustainability. So I want to, just before concluding, I want to say that it's important to have 2 approaches. Bottom-up approaches where you test in the field different projects. So I've been mentioning Prosuli, who was in Botswana, Zimbabwe, and Mozambique. Other projects that are going on. Now we're starting a project in the buffer zone of Limpopo National Park, trying to, in that buffer zone, with human population living in, trying to promote the shared landscape and see what kind of human well-being we can achieve with what kind of conservation objectives. And there are many other people, you will hear about Humani just after me, but also Herding for Health, I'm sure you hear about that, softwood alliances. Lots of people that are testing not only technical innovation, but innovation in governance systems, in trying to promote multi-stakeholder approach and collective action. So those type of projects are very important, but to some extent you need also to have some top-down approach to implement that framework across countries. And so I just want to mention that project, COISA, capacitating One Health in Eastern and Southern Africa. And here we're talking with One Health platform at national level, with Ministry of Health, Ministry of Agriculture, Ministry of Environment. And just to give you an example, so we have 12 countries from Somalia, Ethiopia, down to Mozambique, to Namibia. And 2 weeks ago, we organized a conference in the Wange Safari Lodge, where we had soil health specialists, rangeland health specialists, vets, agronomists, all talking about their own health components and discussing with people from the government, from those 3 ministries, from the One Health platform, and trying to raise awareness and make advocacy so that they integrate more environmental health and ecosystem health within their One Health platform that are currently very much focused on human health and animal health. But so the idea, and the impact will be medium term, long term, but the idea is really to reach those top-down approach with a bottom-up approach in order to really deliver One Health properly. And of course, in Eastern and Southern Africa, Savanna health or healthy savanna is a key component of those because it's one of the main biomes in those 2 regions. So in conclusion, I would say that to promote those healthy savannas, it's a big challenge. And it's about co-designing an idea of savanna for people and for nature. It means that current conservation paradigms need to change. And I repeat what I've said last year, but donors need to change, practitioners, conservation practitioners need to change, researchers, we need to change, and local community also need to change because they've been used to a form of conservation that is not at all sustainable. And I just want to say for the young generation here today, I mean, you need to try to tackle those challenges at the interface because they are crucial and they can have a huge impact. And so just to say that we believe that the One Health approach offers a nice framework for a holistic and system thinking approach to conserve savannas outside protected areas, but also linked to inside protected areas. Thank you.
Thank you so much, Alexander. Unfortunately, you've used all your time, and we will be having the next speaker, Eve, and she will be talking about the elephants in the case of Hangwe National Park in Zimbabwe.
Good morning, everyone. Thank you very much to still be there before the break. So the French continue, and we will speak this morning about the functional of one key species of savanna ecosystem is the elephant and the link between their presence in one ecosystem on infectious disease. So the research question is, could elephant mitigate the risk of interaction among ungulates prone to pathogen spillover to livestock or human in the context of climate change? And it's the case of Wange National Park in Zimbabwe. It's a collaborative work, of course, so I would like to thank all my colleagues. Sorry, I don't know. Yeah, thank you. So thank you also to all the sponsors, to our institute, so from Zimbabwe and also from France and all our networks that it was introduced also by Alexander. So you know that the climate is changing and the African continent where the temperature are rising faster than the global rate will be particularly affected. Climate change could result in the significant losses of African plant species and over 50% of birds and mammal species by 2100. For instance, the population abundance of large herbivore in Africa has declined maybe around 60% between 1917 and 2005. So megafauna-rich ecosystems have been the norm for millions of years, but at the end of Pliocene, half of the megafauna disappeared. So this is the representation of the extinct species in gray in the savanna of South America. So it was really impressive before. So there are lots of publications about the function of the megafauna in literature, about their role on carbon allocation, organic matter, nutrient translocation, seed dispersal, but very few on their function in infectious disease in savanna ecosystem. So here are the representation of the number of megafauna. So we can see that in Eastern Africa or Austral Africa, there are a lot of megafauna still there. But here is the representation of the threatened megafauna. So it's not the case as the Southeast Asia, but also the Eastern Africa or Southern Africa is concerned by potential disparition of megafauna in the future. So as it was mentioned by Virginie also, we know now that 2/3 of the human pathogen have colonized human following an horizontal transfer from animals, so domestic or wild. There are a lot of known cases like HIV, malaria, bovine tuberculosis, influenza. And this is really the core of our research, is about the multi-species transmission, which is a key concept and process in the spread and the distribution of many infectious diseases in animal and human, like rabies, foot and mouth disease, or brucellosis. So we have in the environment frequently some donor species that they can spread their pathogens to recipient species. For example, for rabies virus, the fox is able to infect another fox with one viral particle. But it's possible also that the fox can infect the dog. The dog has the capacity to receive the virus and to amplify it, but with less efficiency compared to the fox. So this is really a concept that is really important for our research. It's about the infectiosity concept. For example, they need to have 1 million more viral particles for infected dog compared to a fox to fox. So this is all the subject about what is the community of infection in the landscape. Little is known on really the mechanism of pathogen transmission among multiple hosts. What are the populations that they are reservoir of infection, breach species, fire breaks, dead animals, and in particularly in empirical conditions, and particularly in savanna ecosystem in Africa. Here, for example, it's a representation of a transmission among disease, and we can see that, for example, for this pathogen that it doesn't exist, but the cat or the dogs can be very important in the maintenance of these pathogens, but also two other species could be less important, but could also be able to transmit the pathogen with less efficiency, but they are still in the system of the community. So during the last decade, literature has suggested that biodiversity alteration could be responsible of disease emergence events. Indeed, wildlife species and extinctions can remove dead hosts belonging to species that cannot transmit, or they are not able to transmit efficiently the pathogen, and therefore dilute the spreading. So I don't know if it's clear about this concept, but if we are back on this representation of a potential transmission disease, if, for example, in the future, these two species disappear from an ecosystem, it means that we have the two species that they are really efficient to transmit these pathogens in the system. And as the nature abhors vacuums, it's potentially also an increase of this population in terms of abundance, and potentially increase the risk of transmission. But at the same time, larger wildlife biodiversity should also yield more parasite species. So it's on the opposite, it's called the amplification effect. So addressing this question on infectious disease dynamic is now urgent on the wildlife. So our model in terms of infectious disease in savanna ecosystem, it's the foot and mouth disease, where it's more or less well known according to the species, but it's known that the African buffalo can be the maintenance host, the maintenance population. It means that they can carry the virus all over the years. And the recipient population could be the cattle with some symptoms in some area and impact on the productions and have economical impact. And we have some data in the literature that other ungulates, smaller ungulates, are able to receive the virus and potentially to transmit, but with more or less efficiency. But what is really important for our study, it's also animals that they are not supposed to be efficient to transmit the pathogen. We call them the firebreak species. For example, like the elephant, the zebra, or the giraffe. They are really important in the ecosystem, potentially for the dynamic of the disease. So we are working in Hwange National Park. It's in Zimbabwe, in the Kaza Transfrontier Conservation Area. And the really particularity of this national park is that it's really porous to the communal area. There is no fence. And also the water is scattered by different water ponds in all of the national park. So the water pan can be pumped or not, but it's not reverse system. So this is wonderful to have a design where we can have the observation of difference in terms of host species, in composition, and also in abundance. For example, we can have a water pan where we can have mainly the reservoir species of the pathogens. So here's a representation of foot and mouth, for example, and we can observe the temporal dynamic, which trains are circulating, and after we can observe if it's a pan where there are the main reservoir species and also the bridge species, what are the trends in terms of dynamics, but also in terms of strengths, and also observe the pan where there are species where they are not supposed to transmit the pathogens. But first, we need to observe the community of all these animals in our ecosystem. So do contact among ungulates vary according to the quality of the rainy season, the vegetation of the presence of mega herbivore in the savanna ecosystem, as savanna elephant, sorry. So we use for that a wonderful